Good afternoon, those in the East, and good morning, those in the West. I'm going to be talking about the work we did as part of this SCRI project on bloom thinning using the pollen tube growth model, what we're calling pollen tube growth model 2.0, and I'll explain in a moment what we are referring to between 1.0 and 2.0. A lot of the modeling work and field validation work was undertaken by a postdoc in my lab, Brent Arnoldson, who's now down at the University of Kentucky. There he is on the far right, doing some hand pollination work in a greenhouse as part of this project. Let's turn on the laser pointer here. So what we were trying to do was to use 10 years of data that was collected, first starting with Ross Byers and then Keith Yoder and his technician, Leon Combs, who worked extensively with the Washington State industry and had a lot of funding from the Washington Tree Fruit Research Commission over the years to develop these pollen tube models. And they were mostly focused for the West Coast, and they were very limited in terms of the number of cultivars that we had empirical data to develop models for. And so it was kind of leaving new cultivars out of the mix and also leaving a large geographic part of the country out of the mix for being able to use this. So in essence, we were trying to add precision to the pollen tube model. So this is familiar to all of you online. Why do we bloom thin? But I'll just review quickly. We know, and Terrence has introduced this, has been introduced many times today, that the earlier that we remove the crop, the larger the fruit size and the greater the return bloom for reducing biennial bearing the next year. So you can do it as fruit buds in the dormant season, and then you have an opportunity again at flowering, and then the more traditional petal fall and fruitlet thinning that we know about. So if we can get in there and bloom thin and remove the crop before the fertilization event has happened and the seeds start to develop, and those seeds start to develop pushing out hormones and gibberellins and such forth that are going to prevent flower bud initiation from occurring, we know that we're going to get better return bloom. Some work that I did in Virginia, we also showed that some of the materials, for example, lime sulfur, and at the time, the formulation that we were using of the biofungicide regalia had fungicidal effects as we did this, so that was kind of nice. And those two materials, again, can be used for organic growers. And so many organic growers can't use the synthetically derived plant growth regulators that are used at the more traditional fruitlet thinning times. And so flower thinning opens up that opportunity for them. And so the pollen tube models was just a way of trying to more precisely make those applications. And as I say here on the negative side, that traditionally the applications were going when approximately 80 or 20 or 80% of full bloom had happened. And so, those are kind of arbitrary numbers. They work to an extent, but not necessarily because of any exact precision in terms of when those are going on. It's just like, let's get them early, let's get them late. We also know that there's risk with flower thinning or bloom thinning in terms of understanding spring frost, and also some of the materials that we're using are caustic, lime sulfur, ammonium thiosulfate, and can lead to russeting issues. So there's some pros and cons with it. So let's just have a quick refresher here on flower anatomy. On the left here is what the apple flower looks like. And of course, this is our king flower. And you can see we have a whorl of smaller flowers, or side blooms is what we call them, on each spur. This is just a cross-sectional view of a flower in a cartoon. And what we're going to be talking about here, we'll talk about really on the far right side, where we take away the petals and the anthers, the male part, and just looking at the female part of the flower here. So this is our stigma on the top. That's where these pollen grains land. They germinate up here. And then once they germinate, a pollen tube, shown here in blue, goes through the style, which is this stalk, shown here in the middle picture in green, until they reach the base of the style and eventually grow into the ovule where they'll fertilize the egg. Okay. So what we're trying to do, and this is the work that Ross Beyer started a couple decades ago at this point, and that we've been working with since then, is model this growth rate. How fast does it take, or how long does it take for these pollen tubes to grow through the style and then fertilize the egg, right? So the time between pollination and fertilization. So the original pollen tube model 1.0, they had all of these trees grown in root bags. They would dig them out, put them into refrigerated truck to get chilling hours, and then put them into a greenhouse to get them to have enough growing degree hours to start to grow. Once the buds started to form, they would then just do what an apple breeder would do and open up the petals, emasculate the flowers, and then hand pollinate. You can see here the brush here with pollen grains on it onto the surface of the stigma. Put them into a growth chamber, different set temperatures, different amounts of time. And once you have time, and then we'd visualize the pollen tubes, and then we'd get length. So here's the visualization process using fluorescence microscopy, and you could actually measure the length of the pollen tube. So if we knew how long they were, we knew how long they were, how many hours they were in the growth chamber, we could figure out the growth rate. So here's, for example, the Fuji pollen tube model at different temperatures, what the growth rate was, and they put that together. Once that was done, then we were able to use data from weather stations, from Ethernet systems, and be able to then use hourly temperature data, plug it into this curve here, and then get the growth rate of the pollen tubes over time. And so this is, again, the models. A couple more have been added since the original work done by Virginia Tech. But basically, here it is in temperature and Fahrenheit and the hourly growth that they had over time. And each of these developed into a model that could be used in either NUA or the Ag WeatherNet system for those in Washington. And so once you started once you had all that data and we had this in, we have this programmed in, this is what the NUA looks like for the Eastern growers. The one at Ag WeatherNet looks a little different, but it's basically the same. The growers would have to measure style length, right? So this is something could be done on the tree, or pick them off the tree and then measure them with calipers. And then this is our 0 point here, where the pollen grains land on the surface. And then at the base here is when fertilization happens. Okay, so that's the idea here is this is pollination here at 0. And then once we get to 100%, that is when it's fertilized. And that's when we would tell growers, hey, put on a caustic spray, lime sulfur, ATS, something else, to kill any more pollen tubes to prevent them from fertilizing the egg. So the idea here is we can set the crop with this first flush of flowers, king flowers, and then put on additional caustic sprays here at like 70% of the length of the style to prevent any more pollen tubes from reaching the ovule and fertilizing the egg. Okay, so it worked mostly. It was a little challenging for growers. Measuring the style length is difficult. And again, the limitation on the number of varieties that are available was an issue. So what we've done is, before I go into that, one of the things that came to mind for me is how often this has to happen in an orchard. And so I've been kind of asking this question, how many pollen tubes does it really take to set a full crop in a high density orchard? So if we had say 1,000 trees in an orchard, just think about that for a second. How many pollen tubes would it take to get a full crop? Okay, so I just did some math on this, and just kind of the back of the napkin, if each flower has 5 carpels, you might have more than 1 seed per carpel, but let's just say we had 1 to develop a full apple, so it wouldn't be lopsided. Say you had 100 apples per tree. I know we're getting more precise with some of the work that others are doing this by 5 seeds, 500 fertilization events are needed per tree. If you had 1,000 trees per acre, right, we're talking about 500,000 or 500,000 pollen tubes per acre. That would be the bare minimum, right? It's going to, to set the full crop. So it's we're trying to model something that's happening repeatedly over and over again in an orchard, and this is kind of a biological process that's happening in mass as opposed to by precision. So anyhow, back to our modeling work, what we did was we looked at the original data from Virginia Tech. We modeled it all out, and mostly looks like those curves that I showed you before. We're starting to see here in that data set, there's a lot of variability. Okay, so this isn't, great when we're trying to do modeling work, and especially if we're trying to look at how do we create universal models and gain precision. This is what all the data look like together, right? If we just kind of glummed all the varieties on top of each other, and you can see what a scattering of growth rates there are at each of the temperatures. So one of the things that Brent did first was really looking at what are the variables that are important for the model. And he quickly realized that using thermal time or growing degree hours is probably going to be a better input than using just temperature alone. And so, and in fact, it increased the modeling potential of our data set, and it was more compatible with field data. Okay, and so he also came up with this new variable, percent styler penetration, which is just as opposed to how fast are the pollen tubes growing, which is what we were trying to do before, looking at when do the actual fertilization event happens. So some of the models that came up with through this SCRI project are a linear regression model, kind of just a simple growing degree hour by the percent styler penetration here, and then for each of the varieties here. And then what's important here is to look at where we are for growing degree hours on the x-axis here. And for a lot of these, we're between 700 and 900 growing degree hours to reach fertilization, right? To reach the styler penetration, as Brent was calling it. He used a logistics model, which actually had better accuracy up to about 90%, which was really nice to see. It's a little harder to use this kind of model in the mesonet systems, but from a biological perspective and just kind of thinking about how this all works, this was a really nice step forward. And we also did some artificial intelligence modeling work using machine learning tools, random forest trees. And I'm not going to go through all the details in here. There's a lot of details. But the important thing is that it really increased the fit of the models. It was able to adapt to the high temperatures a lot better. And it was able to be a better fit for the universal models. The problem with this, what we found from a very practical standpoint, while we liked it from a mathematical standpoint, from a practical standpoint, was that it's really hard to program a machine learning model into the mesonets. So it's something that we're still thinking about and trying to work on, but for now, we just haven't made that jump yet in terms of the computational aspect of it. So for the model 2.0, one of the things is that we can get away from using the style length. You don't have to measure that anymore, which is great. We found that it wasn't, once we started pulling all the data, it wasn't correlated to the pollen tube growth rate. And that we're going to use growing degree hours. And so, and several of these models seem to work pretty well. Like I said, the logistics model has really good accuracy. And another aspect of it is using the linear models, however, is we're able to do some confidence, not exactly confidence intervals, but we can think about them as more like prediction intervals around it. So instead of the model previously telling you needed to spray at Tuesday at 3:30 a.m., the new models are going to say, you've got a range on either side of what we think it is going to be based on some of the more nuanced outputs that we're finding from the models. So this is what it looks like. Here are some of our models in the NUA system that Dan Olmsted helped us program in, the linear, the logistic model, univariate and multivariate. And you can see here the kind of hidden by this text box, but you can see here the orange on top, and then the dashed line down here are the two different confidence intervals or prediction intervals around the line. Okay, so we tried to move this into the field, and the results were mixed. We had some significant fruit set reduction in some trials. We had reduction in seed count, which is promising, right? So we're not fertilizing the egg, but we weren't always able to accurately predict the pollen tube growth rate in the field, right? When we went out into the field and we did controlled pollination and then harvested those flowers and did the microscopy work. We also found that in a lot of years that the amount of pollination was not sufficient, and that really affected the growth rate of the pollen tubes, and also the effect of actually just using bloom thinning as a way of trying to manage crop load. So we kind of went back to this a little bit. We looked at a whole bunch of varieties. This is 2025, just this past summer data that we did. And we just did some controlled pollinations in the field using snowdrift. And we're trying to better understand how many actual growing degree hours are needed for a range of different varieties in field settings. So the modeling work was working with our existing data from those growth chambers, and it fell apart a little bit out in the field. And so it's good. We've got a lot of data we're working on where you kind of, here's all that data pooled together. And what you can see here is that while the data from the growth chambers was showing 700 to 900 growing degree hours, our field data is showing that pollen tubes are really needing more like 1200 on average, but there is some still variety by variety and probably even pollen source, the male part of this variability. So kind of rethinking some of this and really thinking that maybe growth rates aren't as important as just the total growing degree hours, and that by about 1200 growing degree hours, we have fertilization events happening. Not needing to measure styles, I think, is going to be a huge plus for using this. We have some areas that really need some further work and hopefully additional funding through SCRI or other sources, visioning systems to know exactly when should you start these models, and also better understanding flower viability and pollinator activity would really help, right, to understand how many flowers out there are going to actually be able to set an apple, and then also how active are the pollinators. So huge thanks to all the people who worked on this project over the years for the SCRI. There's a lot of other sub-projects that we did, but we don't have time to go into all of them today. And I'll leave it at that and break out of my slides here and see if there's any questions.
Yeah, we got two quick questions, should be good. One is AgWeatherNet going to use the new model this year?
No, we don't have the new model available for AgWeatherNet. I think we're not quite, it's not publicly available on NUA either. We need some more refinement, honestly. And I don't want to release something where we're not confident enough that it's going to work. So we're doing another, we put in a request for some more funding through the Apple Commission here in New York. And hopefully we can get something available for the Washington growers 2027. And we'll work with Tori on that.
Thanks. One more. Could you please comment on mechanical flower thinning?
I have not done any mechanical flower thinning. Others who are on this call have in the past. The Darwin, for example, is a really well-known one. There's a lack of precision with it. And it also can lead to some issues with damaging the tree itself and perhaps be an entry point for fire blight. So there's some concerns with that. I think it has some potential in certain climates and certain types of tree architectures. But anyhow, maybe others who've actually worked on those machines can add some comments into the text box here.
Thanks. One came in from somebody from across the Atlantic. just more of a comment. I think the observation that total hours for pollen tube growth is a key factor, as we have some very cool springs following flowering periods here in Ireland, but still get fertilization.